All right, so in this lecture, we'll talk about pharmacophore-based drug design, PBDD, pharmacophore-based drug design. And this is actually a method of LBDD. LBDD is ligand-based drug design. In many syllabus, what we find is ligand-based drug design is erroneously labeled as pharmacophore-based drug design. Okay, and that is, as I've just mentioned, an error. Okay. So ligand-based drug design is a broader term. So yesterday in our previous lecture, we talked about ligand-based drug design versus structure-based drug design, right? SBDD. And for structure-based drug design, we had taken the examples of docking. We talked about molecular dynamics, right? And as examples of LBDD, we took the name of PBDD, that is pharmacophore-based drug design, and then we talked about QSAR, right? So in this particular lecture, we'll be talking about PBDD, that is pharmacophore-based drug design, which is a example of ligand-based drug design. Okay, so that we have just clarified at the outset. All right, let's go ahead. So pharmacophore-based drug design, a pharmacophore is basically defined as an ensemble, yani a collection of interactive functional groups with a defined geometry. Okay, so this is the definition of pharmacophore. In our lower classes, we have learned the definition of pharmacophore, right? So we talked about all those functional groups and the basic nucleus that is responsible for the activity of the molecule, right? That was our definition then. So we said, for example, again, if we take out the case of naphthalene, maybe, okay. So we are saying that we have naphthalene-based molecules, and then we are substituting them differently at those different positions, right? So after substitution, what we observe is maybe those few of those positions are important for activity. For example, say suppose position number 1, 2, 3, and 4 are important for activity, and the basic nucleus of NAP for activity. Then what would we say? We will say that naphthalene ring substituted at 1, 2, 3, 4 positions with these functional groups is important for activity, right? A classic example is of that of quinolone, right? Quinolone antibacterial agent, right? So that was our definition of pharmacophore then. But now what we are saying is pharmacophore is an ensemble, that is a collection of interactive functional groups with defined geometry. So now we'll not look at the molecules with respect to just those functional groups that is involved in binding interaction with the receptor. We will say that those functional groups which are located at specified geometries with respect to each other. So for instance, here in this particular group, what we see is an aromatic ring fused with another aromatic ring. Okay, so these are two different pharmacophoric points. Okay, these are two different pharmacophoric points, or rather functional groups. Okay, and one of the aromatic ring needs to be substituted at four different positions, right? So maybe this is the electron donating group, maybe this is the electron withdrawing group, maybe this is something like a COH group or some other group, right? And this can be any alkyl group, for example. Okay. So what happens is this is now our pharmacophore. Okay. So we are saying two aromatic groups, one electron withdrawing group, one electron donating group, then one COH group, one alkyl group. And these are located with respect to each other, spatially located with each respect to each other with a defined geometry. Okay. Now that is our pharmacophore. Now imagine once we have this pharmacophore, we can screen, that gives us an a kind of visualization of how the receptor would be, how the receptor would be possibly, right? So we have just mentioned that pharmacophore based drug design is a method of ligand based drug design, and ligand based drug design is used when we know the ligand, right? But we don't know the receptor structure. So in this case, we are using the structure of the ligand, and on the basis of structure of the ligand, we have come up with a pharmacophore, two aromatic rings, one electron donating, one electron withdrawing, one alkyl group, and one COH group. Now, these are pharmacophoric features that are available in the receptor, right? So indirectly, we are talking about the receptor. That the receptor has a binding pocket for electron withdrawing group. It has a big binding pocket for two aromatic groups. It has a binding pocket for electron withdrawing group, a small binding pocket for alkyl group, a small, positively charged pocket for COH binding group, COH functional group, right? So indirectly, what we have done is we have visualized the receptor, right? So that is the reason why pharmacophore-based drug design, which is a method of ligand-based drug design, is a indirect method of drug design, right? Now, once we know these pharmacophoric features, we can screen huge number of molecules. Okay, we can screen huge number of molecules which may have two or more aromatic rings, which may have electron donating, withdrawing, and COH group and alkyl group, or maybe some other substitutions at these positions, which may get into and fit into the receptor's active site. So this, what we have drawn here, is basically receptor's active site. Okay, so even without the knowledge of ligand, what we have done, without the ligand nature, uh, please excuse me, even without the knowledge of the three-dimensional structure of the receptor, we have kind of found out what is the three-dimensional structure of the receptor that is responsible for binding of these kind of ligands. Okay, so now this receptor structure can be used as an hypothesis to check which different type of molecules will be binding to the to this particular hypothetical receptor, and hence we can find out, basically we can mine out, right, we can screen out different structural moieties, different molecules which may fit into this three-dimensional structure, hypothetical structure of the receptor, right? So that is what is pharmacophore-based drug design. So what did we do? We studied large number of ligands, and on the basis of that study, we found out the basic pharmacophore. And now once we know the pharmacophoric features and their geometries with respect to each other, pharmacophoric features and nothing but functional groups. Okay. So once we know that, we'll try and screen large molecules, molecular libraries to find out newer molecules which will fit into this criteria. That they will have maybe two or more aromatic rings fused with each other, and they will have these different functional groups substituted on this particular aromatic group, right? So that is what is pharmacophore based drug design. Now, a pharmacophore may be derived in several ways. One is by analogy to a natural substrate or known ligand. So if we know a particular ligand or a natural substrate for a particular receptor, we can kind of derive a pharmacophore out of it. The other method is by inference from a series of dissimilar biologically active molecules, the so-called active analog approach. So this is what we have just said, right? Now here we talked about structurally similar molecules. We can also use structurally dissimilar molecules. Rather, they are structurally similar but biologically dissimilar. Okay, so few of them would be highly active, few of them would be moderately active, some of them would be less active or maybe inactive. Okay, so that is what is mentioned over here. Dissimilar, biologically active. Okay, and then the third approach is by direct analysis of the structure of a target protein. Now, if we know the structure of a target protein, that would fall into SBDD, right? Structure based drug design. But nowadays, what we do is whatever knowledge we have of the receptor structure or of the ligands which are going to bind to the receptor structure, we tend to use a mixture of approaches. Okay, so even if we know the structure of the target protein, we can always go for SBDD, that is maybe docking or some other method of structure-based drug design, or we can still go for LBDD, that is ligand-based drug design, and specifically we can use pharmacophore-based drug design. So these are the three ways in which pharmacophore may be derived. Now, sorry, most of the pharmacophores tend to be fairly simple. So they may have two or three or four points. Okay, points and a functional group. So in the previous example, we saw this particular example here, right? So here we have one, okay, since both of them are aromatic rings, so we'll consider them as one, then two, then three, four, and this is five. So this will be referred to as 5-point pharmacophore. Okay, this particular example that we took here would be referred to as a 5-point pharmacophore. Okay, but most of the times, the pharmacophores are fairly simple. They may be 2-point or 3-point or 4-point, 4-point and a functional group. Okay, and although some incorporate more elaborate features such as best place, regions of excluded volume, etc. So we can have, those angular geometries and some parts wherein there should be no functional group at all. So that will be referred to as excluded volume. Okay? Then overspecifying a pharmacophoric pattern through the use of restrictive substructure criteria will limit the overall diversity of the identified active molecules. So if we overspecify a pharmacophoric pattern, okay. So for instance, again, let us go back to the previous example. So here we talked of two aryl groups which are connected to each other, rather they're fused to each other in amphetamine ring, right? But if we keep on, specifying more and more details, so if we say that this aromatic ring has to be fused with this aromatic ring, so that means something like biphenyl would be just knocked off, right? So that's the reason why we should not over-specify the pharmacophoric features. Okay. Then, in an ideal pharmacophore, the generality of functional groups does not restrict structural process, while the pharmacophore geometry supplies discriminating power to the search, which means that we should talk about a pharmacophore which should talk about the functional groups, various functional groups that can be accommodated. However, the geometry needs to be fixed. Okay, so if we are saying that this particular naphthalene group, it has to be non-coplanar with this alkyl group, for example. Okay, then that geometry needs to be retained. Okay, we cannot play with the geometry of the pharmacophoric elements. That is what is mentioned over here. So we have to talk about general functional groups. We have large number of functional groups. Whatever functional groups can be accommodated, however, the geometry should be retained. Okay. Now, once a pharmacophore model has been derived, there are in general two ways to identify molecules that share its features. So we can use two different ways to identify newer molecules, newer NCEs which may progress on to become drugs. Okay. So first is by de novo design, which at least in an ideal world generates chemically reasonable novel hypothetical structure. So once we know that we need two aromatic groups that are kind of fused to each other, and then they have to be substituted in those particular mentioned number of ways, then we can think of de novo design. I mean, we can ourselves think of designing, right? So for example, two aromatic rings fused together, उसके बदले we can have two phenyl rings which are simply connected by a single bond, यानी कि biphenyl type of structure. And this biphenyl may be one, two, three, and there may be a substitution either over here or over here, right? Because we need four different substitutions, right? So such kind of de novo design, de novo यानी zero से शुरू करेंगे. Starting from zero, we can design newer molecules. Okay, those molecules can be either hypothetical or they can be chemically reasonable. Chemically reasonable यानी chemically synthesizable molecules. Okay, and then 3D database researching can be done, in which large databases comprising of 3D structures are searched for those matching the pharma. So what we can do is we can use those different databases that are available on the internet. Okay, those databases contain billions of molecules, and those billions of molecules can be screened, and we can try and check as to which molecules may fit into this pharmacophore of two aryl groups together, and all those geometrically, spatially arranged four different functional groups. Okay, so that is how we can use the pharmacophore. Okay, so this is what is pharmacophore based drug design. Either we can do de novo design or we can do 3D database search. However, when we have a single pharmacophore, it is unlikely to recover all the compound, active compound. So this is especially true for antagonists and for enzyme inhibitors. that bind in a number of different ways to block the agonist or substrate binding. Okay. And each structurally distinct class may make its own individual subset of interactions with the total available within the binding site. So whatever space is available within the binding site, each individual functional group, each structurally distinct class may bind to within that space in its own way. Single compounds may also bind to more than one subsite or several different binding modes. They may have several different binding modes. So, for instance, what the thing that we had talked about. So, if you have a small molecule, it might just bind into this area, or it might bind into this area, right? So, what will happen is they will have different binding modes. And given the more stringent requirements of receptor activation, agonists may exhibit less diversity in binding. So antagonists, they may exhibit large diversity in binding. However, agonists, they may kind of exhibit less diversity in binding. Thus, to span the structural diversity and the different modes that are exhibited by antagonists and other ligands, many pharmacophores may be required to be characterized to characterize fully the structural requirements of a given receptor or pharmacologic activity. So we may not have to, we may not be at our best in drug design if we have or if we generate only one pharmacophore. Instead of that, if we generate multiple pharmacophores, so instead of just AR, and 1, 2, 3, 4, we can have something like a simple AR with 1, 2, 3, 4. Okay. Or maybe we can have three AR which are fused to each other with those four different functions. Right. So what we need to do is we need to check check with different number of pharmacophores, and that is how we can end up with good molecules which may go on to become drugs to get approved as drugs. So there is always a need, therefore, to test a reasonable number of molecules that fit into the pharmacophore model. So some of those molecules, as we have just said, some of them may bind to certain parts of the receptor, and we may just get a very bad score for those molecules. But based on the intellect, based on the intuition, based on the experience of the medicinal chemist, such molecules also should be, considered. Many times they need to be considered in order to get active molecules. So 3D database screening, as we had said, will ideally identify compounds with properties outside those of the set of molecules used to define the pharmacophore. So हम लोगों ने naphthalene based molecules use किया था, but when we go for 3D database searching, we might end up getting structurally diverse groups of molecules. Okay, so that allows for identification of novel chemical structures and molecular features, leading to both increased or decreased activity. Okay, so uh, 3D database screening using pharmacophores will lead to identification of novel chemical structures, यानी कि generation of IPR, यानी कि generation of patents. Okay, now this is an example of a pharmacophore. So we have just drawn example of, we have derived an example on the basis of a naphthalene kind of molecule, a hypothetical one, and this is another one. Okay, so look at this. Here we have aromatic ring, two aromatic rings which are linked to each other, and then we have a positive part of the receptor which is connecting with some maybe negatively charged functional group present on the molecule, and a negative charged part of the receptor which is connecting with a positively charged functional group on the molecule. Okay, and these aromatic rings, both of them are having interactions with the other part, the hydrophobic part of the receptor. Okay, so this is our pharmacophoric element. Okay, two pharmacophore, two point based pharmacophore. Okay. Now, when we have to search for molecules, see, have a look. We can have two rings, okay, and they are connected with the CH2 group, and one of the aromatic rings may be substituted, and then we may have two rings with no substitution on the aromatic ring, and then we may have again two aromatic rings. Look at this. This is the phenyl group, and this is the imidazolial group. Okay, now don't bother about the double bonds that have not been shown. Okay, we need to understand that we are talking of aromatic groups. So needless to say, although this is seen as a cyclohexyl group, this is actually a phenyl substitution. Okay, and this also is, it has a nitrogen over here, 5 member nitrogen Na imidazole, right? So see, structural variation. कैसे हुआ? दो phenyl groups था. Now we have one phenyl group and one imidazole group. Now this is supposed to, this will give us IPR, right? This will give us patent because this is a novel molecule. Okay. Now one more addition that has happened over here. यहाँ पे receptor में positive charge है. So we can have something like C double bond or single bond O minus in the molecule, and that will interact with the plus charge in the receptor, right? So see, what have we done? We have done de novo design over here. Two aromatic rings, which is what was expected, and then connected to a functional group which has a negative charge on it. So that will interact with the positively charged part of the receptor. Now, don't you think this is going to generate patents for us, right? So this is IPR, right? And what is also happening is we are increasing the affinity of the molecules that we are designing towards this particular pharmacophore that was defined, right? So the pharmacophore model consists of set of points in space with certain attributed properties and distances between them. These points and their positions define the binding of given groups of ligands with the target. So one thing is for sure that we have two aromatic rings, but they have to have this particular distance between them. So we may as well include two CH2 groups over here, or we may just include one CH2 group over here, or we may try to increase that CH2, CH2, CH2 in the three CH2 groups. But moral of the story is we have to keep these the distances and the geometries between these pharmacophoric features constant. Okay, only then we'll be able to generate molecules which are novel and which are drug-like. So a molecule will either it will fit or it will not fit into the pharmacophore. Now, although a particular compound may fit the pharmacophore reflecting receptor complementarity, its activity is not guaranteed. So what are we trying to do? We are trying to fit in molecules into the pharmacophoric features, right? A molecule might fit in, but it may not have activity. That might also happen. It may possess unfavorable properties such as extreme lipophilicity. ऐसा भी हो सकता है कि some, जैसे नैप्थलीन का एग्जांपल था, उसके बदले if we go on to something like phenanthrene, anthracene, so the number of rings is increasing, the lipophilicity is also increasing, right? So some of the favorable properties may convert into unfavorable properties as well. Likewise, it may penetrate excluded volumes or introduce pairing of like charges. So in this previous slide, what we saw was we had a negatively charged functional group over here and a positive charge in the receptor with which it was complementary. It might just happen that some of the screen molecules may have a positively charged functional group over here. So that will lead to repulsion between the this positively charged functional group and the positively charged functional group on the receptor, right? So such things can happen. Then ranges of activity obtained may have unexpected enhancements as advantageous additional interactions are made with the receptor. It might also happen that we might feel that a particular molecule, it is very slightly binding or maybe it is slightly fitting into the pharmacokinetic feature, but it might just go on to become a blockbuster. Okay, because it might have some unexpected interactions, additional interactions with the receptor, and that might give us enhanced binding and activity in our in vitro and in vivo screen. And a nominal or toy pharmacophore is shown superposed into an example binding site in this particular example. Okay, so that the term that they have used is toy pharmacophore. Okay, instead of using the word illustration, they have used the word toy pharmacophore. Please don't use this word anywhere. Okay, this is just for our understanding. All right. Okay, so ligands found by the search, by the pharmacophoric feature search are of three kinds. Molecules that place other groups into excluded volumes and are as a result weak or inactive. So we can have, those functional groups peeking into the or fitting into the excluded volumes, and these will be weak or inactive. Then the second type of molecules would be those which fit the minimal requirements of the pharmacophore but introduce no extra interaction. So because there are no extra interactions, These may have moderate activity. Okay, these may have moderate activity. And the third type of molecules that fulfill all the pharmacophore requirements and are complementary to the receptor, introducing new groups that are favorable, that have favorable interactions with the binding site. And these kind of molecules will have high affinity. These kind of molecules will have high affinity, and these are the molecules that we're looking out for, right? The molecule of the type 3, which will have additional interactions with the receptor, which are of favorable type, and hence they will have high affinity and hence high activity, right? Examples of successful applications of pharmacophore based drug design. We have pharmacophore models being used for design of many different type of molecules. Okay, so one example is anti-cancer drugs targeting BCL2 family of proteins. So those molecules which target the BCL2 protein as anti-cancer agents have been discovered using pharmacophore based drug design. Many MAO inhibitors have also been discovered using pharmacophore based drug design. Okay, and there are many other examples where pharmacophore based drug design has been combined with other methods of drug design, like for example docking. Okay, so even as a part of my research in MPharm and PhD, I had done such combined studies wherein you know, pharmacophore based drug design is combined either with QSAR or with docking. Okay, and a few other researchers combine it with other methods of drug design as well. Okay, so pharmacophore based drug design is very has been used and it is still very widely used for designing newer molecules. Okay, so in our next lecture we'll talk about docking as a method of structure based drug design. All right, so all of you have just